People always used to say to me, "It's a crazy idea." And I would say, "Well, I know, but I'm going to try."
Once upon a time, there was a girl called Carol. She wasn't that interested in sewing, writing and shorthand, and all the other stuff at the all-girls school she went to, but she had a special gift. She could make things fly, and she really wanted to do something for human health.
I wanted to be able to look at the molecules that my colleagues were making, and the way to do this was to put them into the mass spectrometer. And the mass spectrometer makes them into ions, so it ionizes them, and then it fragments them, and then sort of put the pieces together to give you the original molecule. And I think that whole puzzle-solving thing was something that really captured me from day one.
Molecules are loaded into the mass spectrometer while in solution. By using a thin glass tube, a very fine spray of droplets is created. That's where the magic begins. Molecular ions are emitted and fly through the spectrometer.
We just time the flight, and it's rather like a running race. If you're extremely heavy, it's much harder to compete and come first, so the lighter ones will arrive at the detector first.
Over 2 decades, Carol Robinson has been one of the main driving forces behind developing mass spectrometry as a measurement technique for analyzing small molecules to measuring some of the large macromolecules that interact and decide function in our body. And for this, she's awarded the 2019 Novozymes Prize.
Carol Robinson ending up as the first woman professor of chemistry at Oxford was not a given. Her journey was quite unusual. After leaving school at 16, she joined Pfizer as a technician.
I worked at Pfizer because I knew that I had to be able to get there and back on my own. And I didn't drive at that stage, so I could catch a bus and go to Pfizer. I do remember the interview thinking, I have no idea what I'm coming for.
Carol soon found out she was in her element when she got to the mass spectrometry lab.
Most of the other techniques, you would walk up and put your sample in and then interpret the results. But with mass spectrometry, it was very much get your hands dirty, pull it apart, and have a few electric shocks along the way.
She also started writing in a notebook. One day, one of her supervisors started reading it.
"Oh, this is really good. You should have gone to university." And I said, "Oh, well, it doesn't matter, I'm happy what I'm doing." And then he said, "Well, it's not too late. You could still go to college."
He encouraged her to study part-time for 7 years to get a university degree while still working at Pfizer. Later, she was accepted at the University of Cambridge to do a PhD.
When I got to Cambridge, a new ionization technique had been discovered, so we weren't just restricted to the molecules like aspirin. We could now do peptide chains. So these are the building blocks, the amino acids, and we could work out which one was next to which.
Carol's career was on track, but then something unusual happened. She took a career break for 8 years to spend time at home with her 3 children.
A lot of people said to me, "If you do this, you will not come back." And I remember thinking, well, that's not very good of science if they can't be a bit forgiving and give people a career break. So I remember thinking, well, if they don't take me back, I'll just do something else because I'm not going to be held to ransom like that.
At the age of 35, Carol Robinson returned to science, taking a junior position to work on mass spectrometry in Chris Dobson's group at the University of Oxford. However, the whole field had changed.
While I was out on my career break, electrospray really took off, and this is the whole protein flying, the molecular elephants of John Fenn. So it was a very amazing breakthrough that he made. I just couldn't imagine that you could get a whole protein to fly like that.
Although Fenn got the very large protein molecules to fly, Carol Robinson wanted more, much more. She wanted to use the mass spectrometer to determine the very shape of the protein. She got a crazy idea. if she could spray paint the molecules, she could separate them on the basis of how much paint they were covered by.
So you imagine if it's an unfolded protein, a lot of paint required to coat. If you have a coiled protein, just the surface would be spray painted. And they would weigh differently because deuterium, of course, is heavier than a proton. So I could tell unfolded, heavier than folded.
This method meant that Carol could determine the folding state of a protein. Carol Robinson's scientific colleagues thought she was crazy. At the molecular level, life as we know it occurs in water. In the mass spectrometer, there is a vacuum.
People would say, "Oh, I think your research is made fit for purpose." And I was like, "Oh, you think I made it up?" Or what do you mean by that? I mean, it's really not made fit for purpose. This is what I saw. So that was a hard time.
There was even quite a damning commentary in the very respected journal Proceedings of the National Academy of Sciences.
I did used to send in my papers for review like everybody else and then get back this sort of rather short letter saying, "Well, have you not read this commentary?" I never really doubted because I've been doing mass spec a long time and I knew that the results I was seeing were true, but I doubted sometimes that I would ever convince the field, and I just thought the feeling against me is actually quite strong, so maybe I would be better doing a more conventional line of research which gets published more easily.
In her quest to prove that mass spectrometers can measure folding of even large intact proteins, Carol Robinson realized she could look at even larger complexes like ribosomes and viruses. However, to do that, she had to break the existing mold of what was possible.
We wanted to push the mass to charge ratio on a particular mass filter, and people said, "Well, you won't get any resolution if you do that." But we did. And sometimes I think there's a lot of dogma in the field, and if you actually try something, it sometimes works better than you think.
Carol visited some people she knew at a factory.
I said, "Oh, I'd really like you to build this." And they said, no one's going to use that. I said, "Well, they will. Watch." And could I just have a one-off then of this particular type of instrument? And they said, "Okay, we'll help you design it." And we did iterate a few designs. And then people started to come to the factory and say, "Can I have a Robinson mass spectrometer?" So it became more popular.
Carol Robinson is creative, innovative, and fearless, and she is a role model for all scientists. She went against the dogma and demonstrated that mass spectrometry can be used to measure high mass-to-charge ratio molecules. And today, these mass spectrometers are available throughout the pharmaceutical industry.
Rather than just trying to show that a mass spectrometer did clever things, Carol Robinson now chose to take things to the next level.
I think people thought, "Oh, it's amazing, it stays together," but I don't think they thought it was anything other than a curiosity. I don't think they felt we would learn anything useful. So I think that's really what made me want to discover something for pharmaceuticals or for human health, because most of my career people have said, "Oh, it's very esoteric, it's not particularly useful for anything."
As usual, Carol Robinson aimed high at membrane proteins, the gateways to the cells. At least 40% of all drugs target membrane proteins.
That was quite well established. You could look at the drug and you could work out what it was. But I wanted to look at the drug and its target. In those days, people would say, "Well, that's never going to work, because how are you going to get a membrane protein into a mass spectrometer? You know, this is a crazy idea."
Membrane proteins are incredibly hard to study because one part of the protein exists inside an oily hydrophobic membrane, whereas the parts inside and outside of the cell are hydrophilic.
What we discovered was that these membrane proteins need these protective shells, generally made of detergent. Detergent is effectively soap. So you've made this bubble, and it's now inside the bubble, and it doesn't really mind that you've taken it from solution into vapor. You can have this sort of mesmerizing image of the electrospray spraying bubbles, and then out pops these membrane proteins.
In a series of landmark studies, Carol Robinson unravelled the first spectrum of the proteins synthesizing our cell's energy currency, ATP, and later G-protein coupled membrane receptors, which are targets for many drugs.
Carol Robinson almost single-handed developed a new subfield in mass spectrometry proteomics, and this has allowed for measurement of these large molecules that are important for developing of new drugs as well as for finding new drug targets. This has opened up for a number of new innovations in biotechnology, and for these reasons, she's a very worthy recipient of the 2019 Novozymes Prize.
I always used to think, before I became a scientist, that you had to be some sort of a genius to be a scientist, and I just think there's a lot of people out there like me who just like to create things, like to imagine what's going on in different scenarios, and so I think anyone can really have this career. Don't think you need to be the top of a particular class or table or anything like that. I just think you need to be passionate about what you're doing.
Once upon a time, there was a dame, Carol Robinson.